Hello, pirates. Welcome to our next session of notes. Today's notes is about translation. So the last set of notes we talked about RNA, what it is and what its structure is and how it compares to DNA. And then we started talking about transcription. These notes are about translation. So what is translation? Translation. Well, just like in English, translation means when you're going from one language to another. So in this case, what you are doing is you are changing a sequence of nucleotides to a sequence of amino acids. Okay. And as you might recall, amino acids are the building blocks of protein. So really what we are doing right now is we are having the mRNA that just came out of transcription, and we are turning that mRNA into protein. Now you might see in a lot of textbooks and notes and videos, instead of them saying protein at this point, they might say polypeptide. A polypeptide is simply an unfolded and polypeptide, an unfolded and an cut, unmanipulated sequence of amino acids. So there's no true functionality to it at that point. So you might also see amino acid sequence, but that is what's happening there. So translation is when you go from mRNA to protein. So where does this happen? Okay, it happens outside the nucleus. In the ribosome. Okay, so what is the ribosome? Well, the ribosome is an organelle. It's considered an organelle. It's not a membrane-bound organelle, but it is still an organelle where protein is made. Okay, I'm going to highlight that. The ribosome is an organelle where protein is made. So now that I have my ribosome, here's what ends up happening. So you have your mRNA. mRNA kind of looks like a ribbon. Okay, there's my mRNA. And the mRNA attaches to the ribosome. The ribosome is right here. And remember, the ribosome is mostly made out of rRNA. Okay, but they attach to each other. Then at that point, what happens is you get a molecule of tRNA. And that molecule of tRNA has an amino acid attached to it. So what happens is the tRNA comes in and it attaches itself to the mRNA, and then it drops off the amino acid, and then the whole sequence of mRNA moves over a little bit and it drops off the next amino acid. All right, you don't need to know the process in too much details, but I will go through some of the things. So the first step is the mRNA binds to the ribosome. Okay. The second step is that the tRNA brings in an amino acid. Okay, and here's how it knows which amino acid to bring in. All right, 3 nucleotides on mRNA is called a codon. Okay, those 3 nucleotides on mRNA match 3 codons or 3 nucleotides on tRNA, and that is called one anticodon. So, for example, here's my mRNA. My mRNA here has a codon of CAC. Those are 3 bases. Okay. What's going to happen is my tRNA is going to match up to that CAC using complementary base pair rules. Now remember, this is mRNA and RNA, tRNA, so it does not have the T's, and only has U's. So C would match up with G. A usually would match up with T, but this is RNA, so it matches up with U, and then the C matches up with G. So that's a codon. This here is an anticodon. How I remembered it was the T in anticodon is tRNA. So T in anticodon matches up with the RNA. Okay, the next step, and let me move this down a little bit. Okay, the next step is the tRNA. Oops, step number 3. Drops off the amino acid. Okay, so that's step number 3. And then what happens is the ribosome adds the amino acid to the protein. And then number 5 is the mRNA shifts 1 codon, and the process repeats. Okay, so what happens is you have the mRNA coming in, it gets grabbed by the ribosome, the tRNA anticodon matches up the mRNA codon, drops off an amino acid, then everything shifts over, kind of like an old-fashioned typewriter, if you've ever seen that in the old movies. So here's how this process works. Here's what you guys are going to be expected to do. If I give you a sequence of DNA, let's say my sequence of DNA is G, C, T, and then my next sequence is, let's say, T, A, G, and then A, T, T. Okay, so what you're going to do then is you're going to end up doing transcription to get the mRNA. And the mRNA, remember, complements my DNA. So you directly get your DNA and you turn that DNA into mRNA. So G goes with C, C goes with G, T goes with A, T goes with A goes with U, G goes with C, A goes with U, T goes with A, T goes with A. Okay, then you'll have to remember that these are codons, and there are 3 of them. Here's one codon, here's another codon, here's another codon. Okay, so there's 3 codons right there. So since you have 3 codons, that means you're also going to have 3 amino acids. So now my tRNA anticodons come off of my mRNA codons. So if my tRNA complements my mRNA, I'm going to get G goes with C, C goes with G, U goes with A goes with U, U goes with A, C goes with G, and I'm going to put a comma there because these are separate molecules. U goes with A goes with U, A goes with U. So there's my 3 tRNA anticodons. Now, my protein, or my amino acid sequence, oops, sorry guys. You're going to have to use a translator tool. So you see at the bottom right hand corner how I have that little wheel there? That's going to tell me what amino acids go in my amino acid sequence. The thing you need to know about that tool, and we'll always give you that tool, is that it comes from the mRNA. So what you have to do is see how my mRNA goes into my tRNA. Well, when you do this next part here, you have to go all the way back from the mRNA to get the amino acid sequence. All right, so I'm going to use my highlighter. I'm going to change it a little bit so you can see each one. So let's say here is my first codon. And I'm going to go over to my codon wheel and I'm going to look up and see what amino acid that results in. So to use this wheel, you always start at the center. See where that orange dot is? And then you slide your finger to the first first nucleotide. So I slide it to my C. Then I slide my finger to my next one, which is G. And then I slide it to my last one, which is A. And then I slide it off to get my amino acid. So my first amino acid in my sequence is going to be arginine. Okay, I'm going to put a little bond there. Okay, now I'm going to highlight my next amino acids, or sorry, my next codon. I'm going to highlight that in red. I'm going to go back over here to the wheel again. Once again, I start off in the center. Okay, and then I slide my pen to the first one, which is A, my second one, which is U, and then my third one, which is C, and I get isoleucine. So my second one is going to be isoleucine. Okay, I'm going to put a little bond here. I know I'm a little bit out of the way. My third one then, I'll switch to pink. Oh, undo. My third one, I'll switch to pink highlighter, UAA. I put my finger in the middle of the chart, and then I drag my finger to the first amino acid, or first base, then my second base, first base, then my second base, and then my third base. And notice this time you get a stop codon. So what happens here with a stop codon, you just stop. So it tells to stop making that protein. So you have multiple different types of stop codons. You have one over here, UAA is a stop codon, UAG is a stop codon, and then there's one more over on there it is, UGA. So what you do is you look up these codons and you go all the way on that wheel to translate it into a protein. So what we have here is you have translation, which is when you have a sequence of nucleotides that changes into a sequence of amino acids, which then get turned into a protein. You're going from your mRNA to the protein. It happens in the ribosome. Okay. The mRNA matches up with the tRNA. They complement each other, A with U, U with A, C with G, G with C. All right. And then when you get to a stop codon, the protein stops. Okay. We're going to review this tomorrow in the next set of notes. But right now, I want you guys to practice with the translation, and you guys have some worksheets, some problems to solve, going from mRNA to tRNA, mRNA to protein, and DNA to mRNA. All right, good luck. Don't forget I'm available from 9 to 12 if you have any questions, and I'll see you soon, Pirates.